And now I'd like to introduce our last talk, Dr. John Bell, who is Professor of Medicine at the University of Ottawa. He leads the Canadian Oncolytic Virus Consortium, a Terry Fox Foundation-funded group from across the country that is developing virus-based cancer therapeutics. He also is director of the Biotherapeutics program at the Ontario Institute for Cancer Research. Please welcome Dr. Bell, who's going to talk to us on oncolytic viruses, replicating biological mechanisms for cancer therapy.
So really appreciate the invitation to come speak, and the party last night, John, was amazing. I drank too much and ate too much, so if I nod off during this, just wake me up again. It was a really good party. I've enjoyed the talks. They've been phenomenal. And I think what I want to tell you about today is the work we've been doing with viral therapeutics. And many of the talks you've heard this morning and yesterday I think there'll be parts that really resonate with you. And I think it's an exciting time. I like to think about these viruses, as it says here, as biological machines. And I think we're now at a stage we understand the genetics, the biology of viruses really well. They're very simple organisms, really. And we're beginning to really understand cancer cells a lot better. So I think we can marry these bits of knowledge together and create therapeutic machines that really have a chance to make a big difference. And I think we're just beginning to tap that opportunity. I might just start off by saying that, this shows up here, that my work's been supported for many years by groups like the OICR in Toronto, Terry Fox, CIHR, and many others. And without that support, of course, we wouldn't be anywhere near where we are. And many colleagues who I hope to cite through my talk who have also contributed to this work, and I hope I remember to recognize them all. So, as Dr. Varma said this morning, with the exception perhaps of Lance Armstrong, we don't really do very well with chemotherapy. Our current chemotherapeutic for metastatic disease, is essentially incurable with chemotherapy for the most part. And I think, except for the talks you may hear today from, you heard from Dr. Allison, you can hear from Dr. Wolchuk, this is beginning to change with immunotherapy, but we're still a long ways from getting where we need to be. And part of the reason why we don't cure people with metastatic disease is because this is a picture of a woman undergoing chemo for her breast cancer, is of course that these chemotherapeutics attack not only the tumor, but unfortunately they also attack the normal tissues. And we really get these situations where we can no longer treat the patient anymore because we'll actually kill them with the chemotherapeutic. And so obviously what many of us in this room and around the world have been doing is trying to find what we call targeted therapeutics, things that attack the cancer only and not your normal tissues. And hypothetically, these sorts of therapeutics allow us to continue to treat people until we actually got a good outcome, and we wouldn't damage them from the treatments that we use, either their genetics or their bodies. That's the dream, and that's what we've been trying to do for many years. Our approach has been to use viral therapeutics to see if we can't find a way to make machines that can find the tumor, replicate within them, and destroy them. So we got started this about 10, 12 years ago, and it's really just this picture here I think really outlines what we were thinking at the time. So we thought metastatic disease is something we have to be able to treat with a systemic therapeutic. We wanted to develop viruses that we could administer intravenously that would go everywhere in the body. And the dream was we'd be able to infect tumors wherever they were, but not normal tissues. And I think this picture here really illustrates that well. So this is just a colon cancer model in a mouse. We infuse the tumor into the tail vein, and then it seeds the lungs and elsewhere throughout the body. And then we treat that animal with an intravenous infusion of a virus, which is a replicating competent virus and also expresses GFP. And this allowed us to see where does the virus go? Does it infect the normal tissues or only the tumor tissues, which was what we wanted to do? And how does it behave? And I think what you can see here quite nicely is that when we did this, 24 hours later after infusion, we see that all these white tumor nodules are turning green. So they're productively infected. The GFP is only expressed in infected cells. Interestingly, if you look over here, where there's pink tissue and no tumors, you see that that's also black. So in other words, what we'd hoped to achieve was to get a targeted therapeutic we could give systemically, and it would target all the tumors in the animal, infect them. And in this particular model, we were able to cure all these animals. So you can see in the end, this is obviously a bit of a mocked-up cartoon, but in the end, all the animals would be cured, and we'd see really no sign of the tumor, small little lesions, scar tissue, but that's about it. And then the virus was cleared from the animal. So the oncolytic paradigm is virus goes in, finds the tumors, and grows within them, destroys them, and then it's cleared from the body, and that's the whole therapeutic. So this works great in this model, and if all cancers were like this particular tumor model, which is CT26 colon cancer, which many of you probably work with, well, you know, the problem would be solved. But it clearly doesn't work in all animal models, and it doesn't work in all patients yet. So what we spent the last 10 years doing is really trying to understand why this works here in this system, deconvolute the various aspects of the therapeutic, and then say, well, can we now augment the various parts of it and try to make a therapeutic that will work in more difficult tumors? And the long and short of this, we know now that the most important aspects to this kind of therapeutic are having something that's highly selective, because obviously if it infects normal tissues, it's not going to be as good. Has robust tumor killing. We need to have things that actually productively infect and destroy the tumor. We know that's an incredibly important aspect of this therapy. Tumor vascular attack seems to be an important part, and I'll talk a little bit about that in a minute. And most importantly, I think, the entire field has come to appreciate in the last 3 or 4 years that this oncolytic virus activity stimulates a very potent anti-tumor immune response. And it's this anti-tumor immune response which gives us the long-term cures and allows us to hopefully generate therapeutics that can benefit cancer patients. So the virus I'm going to spend most of my time talking about here, and if I go off track, I'll try to mention it, will be this one called Maraba virus. This is an oncolytic virus that is a rhabdovirus. Rhabdoviruses are very simple viruses. I don't know how many of you are familiar with them, but they are interesting for a cancer therapeutic perspective because they're cytoplasmic in replication. They never enter the nucleus. They just simply enter, they're very simple genetically. They enter the cytoplasm, they replicate themselves very rapidly, and then they bud out and are secreted and pass on to another virus. We were interested in this class of viruses for a variety of reasons. One, of course, is, as I said, they are in the cytoplasmic, so there's no chance to be genotoxic. But they're easy to engineer, they're easy to produce to high levels, and there's lots of reasons why these make potentially great therapeutics. This particular virus was isolated by Dave Stoidel, who was a former postdoc in the lab who went off to start his own research group. And when he was leaving, we contacted Bob Tesh in Texas and said, Bob Tesh had spent his career going around the world and to jungles and so on and collecting samples of viruses everywhere. And his goal was to try to find new emerging pathogens before they emerged. And what he found was not only potential viruses that could cause problems, but a lot of viruses that were fairly benign and really didn't do anything in the human population. So we said to Bob, could you send us your collection? And he sent us about 40 different viruses. And then Dave started sifting through them, looking for ones that actually grew well in tumor cells. And the one we came up with is this one called Maraba. It's an interesting name. It's from a place in Brazil, and its natural host is a sandfly. And so we like Maraba in many ways, and we've done a lot of engineering to make it more potent. And I just want to tell you about some of the concepts that we've developed while developing this as therapeutic. So the first thing I wanted to do is really to discuss the concept that since these are what I would say machines, they have multiple ways to actually attack the cancer. And I think that's an important aspect of a really effective therapeutic. So this is just a cartoon from a review from a colleague of mine a few years back. And so the first point is this, is that the virus we know can infect, and I'll talk a bit more about that, directly into the tumor. As it replicates within the tumor and takes over the tumor's cell machinery, it destroys it. So that's an important aspect, and that's one of the ways it works. The second thing we now know is that the virus is able to attack the tumor vasculature. And this is a very specific event, happens only in tumor vasculature and not in the normal vasculature, and I'll talk about that a bit more. And finally, as I said, the most important thing probably for long-term immune surveillance and long-term cures is the virus is able to infect the tumor. And as it infects the tumor and disrupts it, it releases tumor antigens, but it also reverses the immunosuppressive microenvironment that the tumor has evolved to have. And as a result, the immune system is able to enter into that tumor and begin to have activity against the tumor itself. So I'm going to talk about each of these things and how we're engineering the viruses to be more effective therapeutics. So the first concept is the idea that the virus can selectively infect tumors and not normal tissues. And the talk just before the break with Dr. Balin was actually a great setup for me because it really, I think, helps to explain why it is that we think these viruses can be effective tumor-selective therapeutics. So we all know that as tumors evolve from normal cells, they undergo and acquire a variety of mutations that affect pathways within the tumor. Things like dysregulation of the cell metabolism, as you heard, ability to evade immune detection, really potent angiogenesis. These are all, and this cartoon is not meant to demonstrate it happens that order. These are just things we know happen. And so all these events evolve and shape that tumor to become a more, frankly, malignant entity. What we now know, though, I think from the last 10 or so years of work in my lab, is that the same biological processes that control cell growth and death also control the ability of individual cells to fight virus infections. Okay, so let me explain that a little bit more. all of us are well evolved and engineered to fight virus infections. Every cell in our body has a very important antiviral program, as Dr. Balin mentioned. And this actually involves a lot of genes. About 2,500 gene products get elaborated upon productive infection of a cell. So your healthy cells may encounter a virus, and when they do so, they respond to that virus infection because of their ability to detect the virus invading, and they turn on things like the interferons and all the interferon response genes. And what that does is it actually changes programs within the cell. So normal cells are altruistic. They really care about us, and they don't want us to get sick, so they actually will do things like commit suicide if they get infected to keep themselves from spreading the virus. They'll shut off their own cell division so the virus can't spread. They'll suppress their own metabolism so the virus can't use their machinery to replicate itself. They'll alert the immune system, say, I'm infected, come over here and kill me, get rid of me because there's a virus infection here. And they'll also try to stop blood vessel growth towards them because they don't want that virus to spread. So when you just look at those list of programs, things that normal cells need to do to fight virus infections and are programmed to fight virus infections, those things are all things that cancer cells don't want to do. Cancer cells are not altruistic, so they want to grow forever. They want to never commit apoptosis. They want to actually have accelerated metabolism. They want to find ways to avoid the immune system. So on a programmatic level, cancer cells are shutting down the programs that we use to not only control our growth, but also control our ability to fight virus infections at the single cell level. And so I would argue that the biological processes that underlie antiviral defense mechanisms are really incompatible with efficient tumor evolution. And as you heard from the talk just before the break, many times cancer cells have shut down large components of the interferon response so they can actually do things like avoid the immune system, evade the immune system, and as a result, they also make themselves susceptible to virus infection. And so it's really a deal with the devil that the tumor cell makes. On the one hand, it wants to be able to proliferate and have accelerated metabolism and so on, but in order to do that, it has to give up something, and it gives up its ability to fight virus infections. So at a very basic level, in general, I would argue, cancer cells have found multiple different ways to shut off their antiviral response mechanism so that it can have a growth advantage or hide from the immune system. And for the tumor, that's a pretty good deal to make with the devil because usually the tumor is deep within your body and never is exposed to viruses and infections because we filter things out at our mucosal surfaces and so on. And the concept in the oncolytic virus field is to say, let's make a ton of virus, pharmaceutical grade virus, high levels, infuse it into the patient, and wherever the virus finds a tumor which has got this property, it'll be able to productively infect it and destroy it. So oncolytic viruses are exactly this kind of machine. We attenuate them so that they are unable to overcome the antiviral response of normal cells, but because tumor cells don't have an antiviral response, they can grow there quite well. And that's the basic concept for the selectivity that the viruses we use, essentially all the oncolytic viruses that are being developed now, you really exploit this property. So cancer driver mutations in general, I would argue, are also oncolytic virus driver mutations. Okay, so that's the selectivity. The next thing that's sort of neat about this virus, which was totally unexpected, and we just discovered this by, as John Dick mentioned earlier, sort of serendipity, is that these viruses actually infect and destroy tumor vasculature. And it causes something we call vascular collapse, and we've observed this phenomena in a number of patients we've treated. And so this work started about 6 or 7 years ago in my lab. A really bright graduate student, Jen Patterson, was really trying to understand how do viruses go from the bloodstream into the tumor. And the simple hypothesis was that tumors have disrupted vasculature, and as a result, maybe the virus can just leak into the tumor and initiate infection. But when she did a very close and methodical look at it, she noticed that the blood vessels, which I think you can see here, there's a blood vessel going through the tumor. It is after treatment with a GFP-expressing virus, the endothelial cells that line that blood vessel have turned green. In other words, they've been infected by the virus. So I just finished telling you at great length that tumor cells are more susceptible to virus infection because they don't have an antiviral response. Normal cells have an antiviral response, so they're not susceptible. And now I'm telling you that, in fact, normal cells that line the blood vessels that feed the tumor are infectable. So how is this happening? What's going on here? And actually, just to make the point, these are very, very specific infections, at least in mouse models. This is two really bright students I had in my lab, Carolyn Breitbach and Naomi Silva, who's now a postdoc with Andrew McCart here in Toronto. They set up tumor models, they infected mice with viruses, and they could find that really robust infection of tumor vasculature in a variety of tumor models. But when they took the mice apart, there was no infection of any of the normal tissues especially in the vasculature. So this is a very selective infection that happens within the tumor vasculature and not in the normal vasculature. So what's going on here? So you heard yesterday from Dr. Ferreira and Dr. Dvorak about VEGF, and so it occurred to us perhaps what's happening here is the chronic expression of VEGF within the tumor is somehow sensitizing the normal endothelial cells that line the blood vessels and making them sensitive to virus infection. Is this a possibility? And so this is a typical day in my lab, everybody's sitting out in the sun having a good time. And Roseanne actually, as a postdoc in the lab, had this hypothesis. She said, basically, is it possible that the pathological stimulation through VEGF, through its signaling pathway, is somehow sensitizing tumor vasculature to infection? And so the hypothesis is fairly simple, actually. We know that VEGF activates a defined pathway, interferons act different pathways, but they're both happening within the cell. Is it possible that they're actually cross-talking to each other in some way, and they actually affect each other in some way? And we know that VEGF is pro-angiogenic, and it's well established that interferons are anti-angiogenic. So at a physiological level, they seem to be somewhat at odds. VEGF is known to be immunosuppressive, and interferon is immune stimulatory. We know that interferon's antiviral. Is it possible that because of the networking between these pathways, that in some way VEGF can be virus sensitizing? And that was the idea behind the work I'm just going to show you now. And the thought was that these pathways normally talk to each other in normal wound healing, for instance, and they have to, for reasons maybe both pathways can't be on at the same time. Maybe you want to have inflammation, you want to shut off inflammation, you want to get blood vessel growth, but in the tumor, perhaps this is a pathological situation, and we push over to the one side, and we favor VEGF over interferon. And in fact, this is a very simple experiment to show you here, which is, oh, there it is. So now you're going to be looking at, these are plates of normal human endothelial or mouse endothelial cells, and infected either with a murine version of vaccine that we work with, or another one we've developed, which is called Pexvec now, which is a human oncolytic virus. And again, we're looking again at, in both cases here at human endothelial cells, normal cells. If we treat them with VEGF, murine VEGF, they become sensitized to virus infection. So the virus in this case is engineered to express RFP. And if we take conditioned medium, let me just come back, conditioned medium from tumor cell, which contains VEGF, again, it can sensitize these endothelial cells. So normal cells become infectable by the virus in the presence of VEGF chronically, or in the presence of supernatants from tumor cells which have VEGF. And if we knock out the VEGF signaling pathway, we block the infection. And similarly, if we look on the human side here, human VEGF or supernatants from human cancer cells, they again will sensitize these cells to infection. And we can block that with Avastin, which of course will pull out the VEGF. So as a result, we think we have this and quite a bit more compelling evidence that suggests, in fact, it's possibly true that VEGF soaking of the tumor bed results in downregulation of the interferon response genes in the tumor vasculature and then sensitizes it to virus infection. And in fact, I won't show you all the data, but Roseanne's done a great deal of blotology and microarrays and everything to prove the point. And what she's shown that, in fact, is that VEGF, after it engages the receptor, leads to phosphorylation of STATs, which then turn on PRD1, BF1, also known as BLIMP1, which many of you may be aware of. And this transcriptional repressor then sits on interferon response promoters and prevents the interferon response from being initiated. And as a result, these normal cells now become sensitized to oncolytic virus infection. And we think this is an important aspect of the therapy because we've seen in cancer patients that when we treat them with oncolytic viruses very acutely, within a few days, the tumor itself becomes dark because it can no longer take up contrast agent as a result of what we believe is a vascular shutdown effect. So now the last part I just want to end up with is talking about this concept of these viruses. Not only are they blowing up the tumor, not only are they shutting off and strangling the tumor, but we also think an important aspect is they're really stimulating a potent anti-tumor immune response within the animal. And these are studies which started many years ago with another postdoc in the lab, Kelly, who did a very simple experiment. She took animals, these are immune-competent animals, she put a mouse tumor in them. She then treated them with an oncolytic virus and cured those animals, which at the right model, we can do this essentially 100% of the animals. She waited about 7 months, which is about half the mouse life, and then she challenged them again with the same cancer cell, and of course, they actually rejected it. So they had stimulated an anti-tumor immune response within those animals that recognized that particular tumor. And it's actually interesting, if you use bulb C mice and use different tumors, this will protect against the first tumor, but not the second one. So it looks like a very personalized response against that tumor as a result of the virus infection. And what Kelly did was she did T-cell transfers and all the traditional things you would do in an immune analysis, and showed that it was actually T-cells that were actually generated as a response to the oncolytic virus therapy that led to this anti-tumor activity. And so we thought that was pretty cool, and actually subsequently many labs around the world with different oncolytic virus platforms have shown exactly the same thing, that the act of tumor lysis leads to a stimulation of the anti-tumor immune response. And we see this in patients too, we believe, although it's harder to do the experiment. This is a patient that was treated with one of the viruses that we were developing, a vaccinia virus. It was a melanoma patient, and she had a number of lesions throughout her body, but some on her thigh here. So a few of these lesions were injected with the virus, but not all of them. And then after a while, a few months, we noticed that all the lesions were gone, and in fact they were replaced with what looks like vitiligo. And as any clinician will tell you, vitiligo is a result of your immune system reacting against that part of the skin, and ultimately all the tumors were eliminated. So this oncolytic virus activity is really potent, and it also stimulates anti-tumor activity. The problem is that we see is it doesn't happen in all patients. It happens probably in, I'm estimating from our data and from data of other people around the world, probably 10 to 15% of patients have this sort of a response. So our question is there some way we can change our oncolytic viruses and make it so that this happens more frequently, that more patients respond, and more patients have an anti-tumor immune response? And so how can we make OV therapy stimulate anti-tumor immunity more consistently? And there's many ways you may do it. Our concept was to include a tumor antigen into the oncolytic virus. Okay, so there's lots of tumor antigens out there that are fairly widely expressed. And our concept was, could we actually change an oncolytic virus from just being an oncolytic virus to being an oncolytic vaccine? That is, it acts as an oncolytic virus, but it also stimulates your immune system to recognize the cancer as foreign as well. And then this work that's really been a great team effort with people from McMaster, John Bramson, Young-Hong Wan, Brian Lichty, Byron Brittle, who's now at University of Guelph, and Dave and I in Ottawa. And so the concept here, this is just from an article which we recently published in Nature Cancer Reviews, is as we all know, you can make a cancer vaccine. It depends what antigens you want to use. And you do this by really injecting a vaccine agent into the muscle and stimulate an anti-vaccine response. And that, unfortunately, hasn't worked very well, but you can do it, and you can demonstrate that you actually generate immune responses. And this is because you locally stimulate the immune system, or in the peripheral. We also know that oncolytic virus therapies are able to sometimes enter tumors, disrupt the tumor, and the draining lymph nodes, for instance, pick up the tumor antigens, and you again generate a local response that can potentially really have activity against the tumor. And our thought was, why don't we just combine those two things together? We're going to give one therapy anyhow. Why don't we put something that would stimulate the immune system peripherally and also give us this in situ vaccine effect? And that's what we've done. And I'm pretty excited about the results, and I think this is something that's going to be a really interesting new approach in the clinic. So the way we do this is basically we reprogram the cancer patient's immune system. We give them, in this case, for the purposes of proving the concept, we give them a priming vaccine. And we often use adenovirus, although any kind of vaccine will work. It could be a listeria vaccine. It could be a pre-existing immune response. It doesn't really matter. It turns out that it's this boosting effect of the oncolytic virus, which is really where the magic happens. And so we include, in our experiments, we prime with an adenovirus vector. We then boost with an oncolytic virus, both of them expressing the same tumor antigen. Because we use two different virus vectors, the immune system sort of ignores the vectors, and it focuses activity on the tumor-associated antigens. This heterologous prime boost is really important. So this really is a very profound, and we think is probably one of the most profound cancer vaccines that's ever been developed. We see results which are really much more potent than any vaccine that we've looked at in the literature. So here's a simple experiment. This is a B16 lung model. B16 is really infectable by oncolytic viruses, but not treatable. That we can infect them, but essentially that we can't cure them with the virus on its own. But if we now take B16 and we prime the animals after they've got the tumor, we prime them with an adenovirus expressing DCT, which is a melanoma antigen, and then we boost them several days later, about a week later, with a Maraba oncolytic virus, again expressing DCT, a soft melanoma antigen. And then we examine what are the responses in T cells. We see that in these animals, we're getting anywhere around 30 to 50% of all the T cells in the periphery are now reactive against the self-antigen, which is really quite a remarkable level. And I won't go into the details, but we understand the biology behind this now, and it's a completely unique biology that this virus has, and I think it's something we're going to be able to really exploit. The other neat thing about this approach is not only does it generate really potent and high-level T-cell responses, but it also causes recruitment of the T cells into the tumor. So it's not just enough to make a T cell, you got to get it into the tumor. And if we look at TIL cells, if we use the prime and then boost response, we see we get tremendous infiltration of functional anti-tumor T cells into the tumor. And we don't get that if we don't have the tumor antigen in both vectors. And the second thing we see is we now see what was called epitope spreading, which means that now instead of just the virus, not just the antigen that we vaccinate with, but other antigens found within that tumor, the immune system begins to recognize. So we sort of light the fire with the virus and this vaccine effect, and then the immune system broadens its response against other antigens within the tumor. So we now have taken this and done a lot of work, including non-human primate studies, which I don't have time to talk about today, that are really compelling and convince us that, in fact, this is an approach worth testing in people. And we have a phase 1/2 trial, which is just about to start next week in Ottawa. It's also going to be in Toronto, Hamilton, and Vancouver. And this is a trial that was designed by Neil, Janet, and Derek, who are clinicians working on this trial with us. And the concept really is to use, first, in one arm, the oncolytic virus on its own with a TAA, a vaccine on its own, and then combine these into a dose escalation and ultimately a phase 2 extension. So that trial is opening next week, as I said, and we're really looking forward to seeing what sort of responses we get in that trial. So I just want to end by saying that I really want to try to convince you that oncolytic viruses are an important new therapeutic that we should really work on more. The young people here, I want you to start thinking about it a bit more, because I think they're very cool machines that we can make do the tricks we want. They're cytolytic, so they directly infect and destroy the tumor. We have this concept of self-amplifying, so the virus goes in, makes more of itself as time goes on, so it's a unique kind of therapeutic in that perspective. It can be a gene delivery vehicle. In our case, we use the Tumour Antigen as a gene to be delivered by the virus, but you can imagine putting in cytokines, checkpoint inhibitor antibodies, and these sorts of things, which we're on doing. It's a systemic therapeutic, so we can give it intravenously. It can target metastatic disease wherever it is. It's a sensitizing agent. We know that it actually makes tumors more responsive to chemotherapy. Vascular targeting, we have good evidence both in animal models and in people that these viruses infect tumor vasculature. It obviously induces inflammation, and it's local inflammation within the tumor bed is a really important aspect in trying to generate immune responses. And so that's why I think there's lots of other possibilities to this platform as well, which creative minds could come up with. So we've been able to do this work in large part because we've been supported by a lot of people and organizations, including the OICR here in Toronto, which helped us get our manufacturing suite in play. And now we have a setup in Ottawa where we can discover things in the lab. We have a manufacturing capacity. We can make the viruses ourselves as pharmaceutical grade. And we have great cancer centers in Canada where we can test these. And I think this sort of iterative development is really suited for a biological platform like a virus, and something we're really trying to push forward much faster. And so really, a lot of this work was done, I get to talk about it, but it's done by a lot of different people from across Canada who work together with us in the team, partly through the Terry Fox program grant, partly through what we get here in Ontario. And I'd really like to acknowledge those people. So thanks very much.